This is a video for Year 9 for their revision for their Sense Point 4 at the end of this year, based on their new Edexcel GCSE for Science. So this video is going to go through the biology topics that you need to know, so I'll quickly go through those. There'll be two more videos, one for chemistry and one for physics. So the first topic you need to know about is cells. There are two types of these. There's animal cells that are on the left and plant cells that are on the right here. So both these cells have a nucleus, they have a cytoplasm and a cell membrane. Know the jobs of each of these. So the nucleus controls the activity of the cell, cytoplasm is where the chemical reactions take place, and the cell membranes control what goes in and out. The cell wall, just in a plant cell, strengthens the cell, and the vacuole is filled with cell sap to help the cell retain its shape. Again, that's only in a plant cell. Chloroplasts, they absorb light energy to make food, and chloroplasts are only present in plant cells. Remember, the nucleus also contains genetic information, so that's the DNA, and plant cells have a cell wall which is made of cellulose and a vacuole full of cell sap, which is a solution of sugars and salt. Oops, it was dropped too far down. So, another type of cell that you need to know about is called a bacterial cell. So bacterial cells are full of chromosomal DNA, so it's this nuclear material over here, so chromosomal DNA. And they have one long looped chromosome that controls cell activities and cell replication. The bacterial cells also have no nucleus, so that's what this nuclear material floating around is. And they're these plasmids of small loops of extra DNA that are not part of chromosomes. They contain the extra information, so like genes for drug resistance and the ability to create insulin, so all of that's within the plasmid. And they can be passed between bacteria. So that's how they actually do genetic engineering of insulin, by using that idea. Okay. The flagellum, just at the end over here, is a long hair-like structure. It rotates, allowing the bacteria to move. So it acts sort of like a propeller. It spins, allowing the bacteria to move. The bacterial cell also has a cell wall for support. Cells have been studied for a long time using light microscopes, but only since the 1590s have we been able to magnify images so we can actually see the internal structures inside of them. Stuff like the nuclei, chloroplasts, and mitochondria. Magnification can be calculated by dividing the length of an image by the length of the specimen. Let me show you that equation. Okay, so magnification is length of the image divided by length of the specimen. Make sure you know that equation. Microscopes have changed since the 1590s. So in 1930, the electron microscope was invented. That allows scientists to observe smaller structures in more detail. Okay, so stuff like plasmids and also the internal structure. You can see there's more detail. So some cells are specialised for their job. They have specific features. So sperm cells have an acrosome as part of the head, which is enzymes which digest the egg membrane. They also have a haploid nucleus, which means they only have half the information for a human, 23 chromosomes. They also have many mitochondria to provide energy and a tail to move towards the egg cell. Egg cells are also specialised for their job. So egg cells contain nutrients in cytoplasm, which is required for mitosis. So nutrients which they need for mitosis. And that allows the cells to replicate, and they have a haploid nucleus, yet again to have half the information for a human, 23 chromosomes. And they have changes in the cell membrane after fertilization to protect the fertilized egg and prevent entry of other sperms. Ciliated epithelial cells contain cilia that are like tiny hair structures on the back of the surface of the cell. They sweep hair, mucus, trap dust and bacteria up to the back of the throat where it can be swallowed. So when magnifying cells, there might be certain distances that I mentioned. These are listed here. So milli is times 10 to the minus 3, so it's the same as divide by 1,000. Micro times 10 to the minus 6 divided by a million. Nano times 10 to the minus 9 divided by 100 million. And pico times 10 to the minus 12, so 1 with 12 zeros. So that number at the top tells the number of zeros. Right. Let's move on to enzymes. So enzymes are biological catalysts. Okay, they make chemical reactions happen faster, including things like DNA replication or protein synthesis or digestion. and they are folded into complex shapes that allow smaller molecules to fit into them. There's the idea of a lock and key hypothesis. So in other words, only certain substrates can match with certain enzymes. Only certain substrates match with certain enzymes, a lock and key. So when that match happens, it causes the chemical reaction to happen. It lowers the energy required for the reaction to take place, and therefore it happens a lot faster. The reaction happens a lot faster, therefore you can break down food quicker. You can actually change the shape of an enzyme. That's a bad idea because it means the enzyme won't be able to do its job properly. And we call that change of shape denaturing. So when enzymes denature, they can't work properly. High temperature can do this, extreme pH levels can do this as well. We don't say the enzymes have been killed, we say they've been denatured. So digestive enzymes can break down carbohydrates into sugars, proteins into amino acids, and lipids into fatty acids and glycerol. So there are some factors that actually affect the enzyme intensity. Okay, so these affect how quickly, enzyme activity. These affect how quickly the enzyme actually gets to work. So as temperature increases, so does the rate of reaction, so does the rate of enzyme activity. But high temperatures denature the enzymes, just like before, just like I mentioned before. So here's a graph for it. You can see it increases up to about body temperature, faster than enzyme activity, then that denaturing occurs. Similarly with pH, there's an increase in enzyme activity to about 8 pH, and it drops normally between 7.5 and 8, but it does vary for different situations and different enzymes. Yeah, again, there's denaturing happening either side of that optimum rate, that optimum pH in the middle there. The amount of substrate you have also makes a difference. So the more substrate, the faster rate of reaction, up to a point where it's saturated. In other words, no more substrate can actually get to the enzyme anymore. So there's no more keys to be opened by that lock. All your keys are in use. Yeah, that's the idea. Or no more locks to be opened by the key. Every single lock is full. So there's spare locks that are just lying around, no more keys left to fill them. So you can investigate the rate of enzyme reactions by sort of investigating how starch reacts with amylase, and you can read through this, sort of go to this experiment. You can see that over time it will start breaking down, there'll be no more starch present. So diffusion. Diffusion is where particles go from an area of high concentration to an area of lower concentration. So diffusion is where particles go from an area of high concentration to an area of lower concentration. So when respiration takes place inside the body, oxygen and glucose are being used up, so that means less oxygen and glucose in the cells, there's more in the blood, so it goes from the blood to the cells, higher to lower. The greater the difference in concentration, the faster the rate of diffusion. Osmosis is a diffusion of water, so it goes from where there's a high concentration of water to where there's a low concentration of water. This normally happens through a partially assembled membrane, and that only allows the molecules, smaller molecules through, like the water molecules. It's very much like a sieve. It only allows certain size particles through. This is how water moves in and out of cells. Osmosis occurs as there's a difference in concentration inside and outside the cell. The more water in a substance, the more dilute it is. Active transport is where root hair cells absorb minerals from the soil. In this situation, you need to go against the concentration gradient. So the root hair cells are taking it from an area of low concentration to high concentration. That's called active transport. Energy is required for this process. And this process is also essential for growth. While we're on the process of growth, it's a good time to talk about mitosis. There's a few key words you need to know about mitosis. The haploid number of chromosomes is 23 chromosomes. A diploid number, that's 46 chromosomes. A haplete gamete will therefore contain 23 chromosomes. So mitosis is the production of two daughter cells, each identical sets of chromosomes in the nucleus of the parent cell. And this results in the formation of two genetically identical diploid body cells. So in other words, you have one body cell and you make an exact clone of that, so you end up with two identical body cells. This occurs during growth, during repair, asexual reproduction as well. Cloning is an example of asexual reproduction that produces genetically identical copies. So the phases you need to know are interphase, prophase, metaphase, anaphase, and telophase. So interphase, prophase, metaphase, anaphase, and telophase. Sorry, you also need to know about cytokinesis as well, which is here. Cytokinesis is where the two daughter cells a form with each of which 46 chromosomes. So feel free to pause the video here, work your way through this diagram to make sure you understand each stage and what's going on. Cancers are often caused by part of this process due to that uncontrolled cell division that's taking place there. So growth and development. Okay, so there's a lot of notes here that you just need to read through. I can just go through a few bits with you as well. Growth is defined as an increase in size, length and mass. So wet mass is mass containing water, and dry mass is mass of an organism with no water within it. So three processes take place whenever growth is happening. So cell differentiation, so cells are becoming specialized. Cell division, so mitosis, just like we've discussed. But also cell elongation, where a cell expands, making the cell bigger, so making the plant grow. All three of these processes above happen in plants, but only cell differentiation and cell division happen in animals. You could be asked to analyze data, which could sort of show age against growth, and you'll need to analyze what's going on there. So make sure you've brushed up on your data analysis skills. So stem cells. Stem cells you did about right at the start of year 9. The stem cells are found in the embryo, and they're unspecialized cells. In other words, they do not have a job. They have the potential to grow into any other type of cell. So we call that idea of growing to have a job differentiate. Okay, so they differentiate to have a job. So in animals, they're not usually made until adulthood. They're not usually produced into adulthood. So usually they come from embryonic stem cells for research. That's why there's a lot of controversy, because we have to get these stem cells from embryos. Stem cells that come from aborted embryos or spare embryos from fertility treatment. Some people think this is unethical because that could have been a life. But the problem is that stem cells could be used to cure things like Parkinson's disease, and rebuild bones and cartilage, repair the immune system. So it's a bit of an ethical issue. We don't know whether we should be doing this or not. It's not a decision that just scientists can make. Many people need to be involved. Plant cells make stem cells all the time, so it's not really an issue with plant cells. In a plant, only stem cells are reproduced, and they travel to a specific place, and then they differentiate there. Right, so moving on to the final topic, which is about the nervous system. Apologies for keep hitting that button. So, neurons. Neurons are nerve cells, and they transmit information as electrical impulses around the body. They're branched endings, so you can see them over here. And they're called dendrons. And this is a dendrite here, so it's dendrons. They have dendrites on them. The dendrons can connect with lots of the neurons. The electrical impulse travels along the axon, so that's a bit down the middle, and it's insulated by a myelin sheath. Please use the phrase insulated by a myelin sheath. This stops signals being lost and also speeds up the impulse. Neurons are long, which is better than having lots of little neurons, as travelling between neurons slows an impulse down. The connection between two neurons is called a synapse. So you see a little synapse over here, where it connects between two, and you can see they're zoomed in here. Okay, so if we're connecting between two neurons, we call that a synapse. So what happens is the electrical signal comes down, it becomes a chemical signal, and then an electrical signal again on the other side. So differences between nerves and hormones are: nerves are fast, and while hormones are slow. Nerves act for a short amount of time in a precise area, while hormones act for a long time in a more general area. And nerves transmit electrical signals, while hormones are chemical signals. So we're going to talk very quickly about the structure of the central nervous system. So dendrons pick up a chemical signal from another neuron and convert it to an electrical signal. Axons conduct it, just like I mentioned, insulated by a myelin sheath. The central nervous system consists of the brain and the spinal cord, and is linked to the sense organs by nerves. So nerves link the CNS, the central nervous system, to the sense organs via the brain and spinal cord. The stimulation of receptors, so that in the sense organs, so cells in the sense organs, sends electrical impulses along neurons. The 5 senses that contain receptors are in your eyes, ears, nose, mouth, and tongue. Eyes, ears, nose, tongue, and mouth. They have long dendrons and short axons that carry nerve impulses from the receptors into the sense organs of the central nervous system. There's relay neurons there with many short dendrons and axons that carry impulses from the sensory neurons to motor neurons. Motor neurons carry several nerve impulses from the central nervous system to the effectors. So when you actually have a reflex, you feel the pain in the finger, the sensory neuron then carries that information to spinal cord. The spinal cord realizes this is a reflex action that's required here and sends that along with relay neuron to the motor neuron. The motor neuron carries the impulse to the spinal cord back to the muscle. The muscle then bends the arm because of the signal being given to it, the muscle there being an effector. It pulls the arm up so you don't actually feel pain in your finger. Same thing when shining light into eyes, that's why eyes squint, because the stimulus is the light going into the eye. The sensory neuron picks it up, sends information to the relay neuron sends it to the motor neuron, then to the muscle, which makes your eye close. So that there is the reflex arc. And that is the end of the biology video for CP4 for Year 9 GCSE Science.